Hi, this is Miss Litton, and this is AP Biology 7th period review of Chapter 13, Regulation of Gene Expression. Say hi. Hi. All right. Did you know it's going to rain today?
Yes.
Are you going to pray for the hills to stay up on the hills?
Yes.
I'm so scared about that. All right. So, key thing we need to understand, big ticket items. Hats and hoodies are off because you're so respectful. So what we need to remember is that the DNA is the same in every single cell in your body, right? Yet different genes are turned on in different tissues and at different times. It could be cycling within hours, it could be cycling within a single day, it could be within months, it could be within years. So why is it that certain genes are turned on and other ones are turned off? And that has to do with differential gene expression. And we first talked about like all the areas where control could take place, but this is an example of what kind of cell, a prokaryotic cell or a eukaryotic cell? Eukaryotic. Yeah, how do you know it's a eukaryotic cell? It has a nucleus. Good, it has a nucleus. So if we were going to look at the 5 levels, we could be looking at the chromatin or the DNA, right? Whether it's heterochromatin or which one is inactive? Because it's dark staining. What's an example of heterochromatin? R bodies. Good. Then transcriptional control, and this is whether or not RNA polymerase binds to the promoter. Sometimes something can get in its way. What's something that could get in its way? Repressor could get in its way, or maybe it doesn't have help. What kind of help could it have? Okay, sure. But that would affect the repressor. I'm thinking of something else. Enzyme? Starts with a T, 2 words. Transcription factors that might need help to bind onto it. Okay. Remember, eukaryotic cells, it's more about positive control, which has to do with transcription factors and activators. Whereas in prokaryotes, it's more about negative control, which has to do with what? Repressors. Okay. And I'll review inducible and repressible. I'm assuming you want to talk about that. Okay? So transcriptional control is the biggest area of control. What's another? Now let's go one more out. What's the next one out? Post-transcriptional control. What does that have to do with? Cap, tail, cutting out the introns. So it's what combination of exons you have and your ability to what? Leave the nucleus. That's all post-transcriptional. The first 3 really occur within a nucleus when you're talking about a eukaryotic cell. The next 2 occur outside of the nucleus. What's the next one? Translational control. Can it get on a ribosome? Can it be translated? Is there a small interfering RNA that's binding to your messenger RNA that disables the tRNA from landing on a codon? Does that make all sense? Yeah. Okay. So that's all translational control. Then what's the last one? Post-translation. Because just because you have a string of amino acids, it doesn't mean it's functional unless it's what? Folded. Who folds it? Right. So you have to have it folded into the correct shape. So all those are areas of control. So then we started off, let's talk simply, we talked about prokaryotes. And this was just so we could learn about the operon, which is what you would find in a prokaryote to control a whole sequence of genes relating to one metabolic activity usually. And prokaryotes are not going to make a protein for something they don't need, right? So they're going to wait until they need that in order to form that. So in this case, we just learned the basics. Regulator genes, notice this is / right here. This means it could be far upstream. It's not part of the operon at all. Can it impact the operon? Sure, but it's not part of it. Regulator genes code for repressors. Sometimes those repressors are formed, and as soon as they're formed, they are good to go. They're ready to bind to a operator, okay? And those are then inducible operons because you have to induce the repressor away, right? Sometimes repressors, when they are made, transcribed and translated from the regulator gene, they're not functional. They need a co-repressor to become activated. Okay, some of you are like, no, I get it. The co-repressor activates that repressor, right? And so it will then go on and stop it. That's called a repressible, right? Repressible because it's not currently being repressed, but if you have a co-repressor, it will repress it. That's a repressible operon. RNA polymerase always wants to bind to the promoter, It's just a matter of whether the repressor is bound to the operator, which neighbors the promoter, right? If RNA polymerase would get on the promoter if it could. But if a repressor is on the operator, it blocks it because it kind of spills out over to it. So either we need to do something in this case to remove this active repressor. We studied the Lac Operon and the Trip Operon. Now, Lac Operon had to do with bacterial cells wanting to break down lactose for nutrients. Now, that's not their first choice, though, right? What's their first choice? Glucose. Glucose is their first choice. But if there's no glucose available and lactose is there, lactose acts as the what? Inducer. Lactose acts as the inducer. The lactose will actually bind to, if you have a repressor on your operator, the lactose, if you have a bunch of it, will bind to the repressor and act as an inducer and remove the repressor so that RNA polymerase can bind to the promoter. If RNA polymerase binds to the promoter, it's going to start transcribing the structural genes. Those structural genes, once they are transcribed and translated, will do what?
Make proteins.
That do what?
That break down.
Yes, that digest lactose, including the lactose that is currently inducing the repressor away. So once all the lactose is gone, we don't need to make those anyway, so that makes sense. Then the repressor gets back on the operator. Who coded for the repressor? Regulator gene codes for the repressor. What does the promoter do? What's it there for? Yeah, it's where RNA polymerase binds. And these structural genes are what you want. Okay, look at me. Questions, concerns on an inducible operon? Yes?
So would this type of control be negative control?
Yes. And you know why it's negative control? Because you're dealing with, just in this picture, you're only dealing with a repressor. Repressor could be active or not active. Now you know because we leveled it up, right? We leveled it up because we know about CAP, right? And cyclic AMP, right? So we included also positive control later on in our discussions. Yes?
In this case for the Lac Operon, is the repressor more active or?
Yeah. It is. This is what's called an inducible operon. The repressor is ready to go. And lactose, and this is usually true in catabolic reactions where you're going to break something down, you need an inducer. You need something to trigger it. You know, I don't want to make anything to break it down unless I have the thing to break it down. Does that make sense? Okay, what else? Okay, so we learned about the Lac Operon, and then we also learned about the trip. Okay, and in this case, you have an inactive repressor. And so when it's formed, the regulator gene is transcribed into mRNA, it's translated into a protein. That protein is an inactive repressor. And RNA polymerase has no problem getting on the promoter. It transcribes the structural genes. Those structural genes are translated into proteins that will ultimately synthesize tryptophan. So this is not a catabolic pathway, this is a anabolic pathway. So as tryptophan increases and we have enough, we need a way to turn it off. Like, okay, stop, we've made enough. So tryptophan acts as the shutoff switch. Like, stop making, I have enough. It's a way to let you know and communicate. Don't waste any more enzymes, energy, or amino acids. So tryptophan, okay, in this case acts as a what? Co-repressor. And it takes the inactive repressor and activates it so you shut it down. And it'll stay shut down until the levels of tryptophan what? Fall below what you need to maintain. And then it'll switch it back on again, right? Because there won't be any tryptophan there. And so the repressor becomes inactive again. So this is what's called a repressible operon. It's usually on. This is part of an anabolic pathway. You're building, building, building until I have enough. Stop, right? And the other one is for catabolic pathways. I don't need to make the thing to break it down unless I have actually something to break down. If you make it make sense in your head, then you don't have to do any memorization, right? Okay. So look to me. Is there anything you need me to go over on the trip operon that you want me to talk about? Okay. Tell the person you're sitting next to right now, of those two, which do you feel more comfortable with and why, or do you feel equally comfortable with both? Go ahead. come back to me. Was there anything you want to ask me now that you just thought of when you were talking with your bio buddy? Anything? Anything you want to ask? Yes.
Just for clarification.
Yes.
With the TRP Operon.
Yes.
In this case, it's positive control and it's anabolic because.
No positive. Anytime you're just dealing with a repressor, whether it's active or not, is negative control.
Okay. So the TRP Operon is negative control?
Anytime you are dealing, both of these had to do with whether repressors were active or, oh, not that one, dang it. Both of these were just dealing with whether repressors were active or not active. And anytime you're dealing with a repressor, repress you, that's very negative. I'm repressing you.
Oh, okay.
That's negative control. If you're dealing with something that's helping RNA polymerase bind to the promoter, a transcription factor, that's positive control.
Okay, so anything that prevents it from binding?
Anything, whether or not you have a transcription factor active or not active, anything it's doing with RNA polymerase activating it is positive. Anything doing with any kind of repressor is negative. Prokaryotes usually live in the negative control regions. Our eukaryotic DNA is usually about positive control. Anything else? Okay, so then we leveled up, right? Because then we remembered that prokaryotes would rather have glucose to eat than lactose to eat. So there's a way to ensure that. And that's because when glucose levels are low, when you're running out of glucose, then you have cyclic AMP. Remember that part? Okay. So at camp they ate all the glucose, so it's low. I don't know. Okay. Glucose is low. Okay. Camp is high. So when we looked at CAP is something, a transcription factor, which will activate and help RNA polymerase bind to the promoter. But CAP only becomes active when you have cyclic AMP. When will you have cyclic AMP? When you, yeah. But RNA polymerase is still not going to get on that promoter if there's a repressor there. Why would there be a repressor there regardless of the state of our lactose? Why would there be a repressor there? There's no lactose. So you have to meet two conditions. No lactose will get rid of the repressor, right? I'm sorry, let me say that again. If you have lactose present, you can get rid of the repressor, right? So if you want to get that repressor out there, you better have some lactose. But you've got to have, if you don't, you've got to have glucose absent in order to activate your CAP with cyclic AMP. So you have to have both conditions. Absence of glucose, presence of lactose. Presence of lactose will get rid of the repressor. Absence of glucose will give you what? Cyclic AMP, which will activate CAP, which will activate or help RNA polymerase what? Bind to the promoter. You got that? Two conditions. Presence of lactose to get rid of the repressor, absence of glucose to help RNA polymerase bind to the promoter. Okay? And we went through several different scenarios. We looked at these different scenarios where we said, okay, when is it you actually are going to transcribe it? Okay, questions you want to ask me about that? Yes?
So if glucose and lactose are present at the same time, then would it not copy the...
Right, so let's talk about that. Lactose is present. Lactose is present, so the repressor is gone, right? But RNA polymerase doesn't bind well because there's still glucose present. Yeah. So it would bind very weakly. You wouldn't get a lot of that enzyme because it wants to focus its energies on the glucose. What else? You good? All right. Then we jump to levels of control, and we reviewed this right at the beginning, right? Each of these 5 levels of control. And basically, you just want to have an example of each one of these levels of control. If I ever asked you an essay about it on your final, I could say, tell me the 5 levels and an example of each, right? And you could do that for me. So you could talk about how DNA is wound on those histone proteins. Remember, we discussed epigenetic inheritance, things above our genes, things that might be in our environment that will inhibit DNA from unwinding. We talked about methyl groups. Do you remember that? Okay, so all of these things things will affect whether DNA is available for transcription or not. Then, and we talked about heterochromatin and calico cats and all. Then we talked about transcriptional control. This is where most control is influenced, is right here at transcriptional control. Do you have an active transcription factor to help RNA polymerase bind to the promoter? Is there a repressor that's in the way? And we talked about the difference in eukaryotic cells. It's all about mostly positive control. You have to have this, this, and this, and this before you have enough things to help RNA polymerase bind to the promoter. We talked about another binding site, the enhancer site, and these activators bind to the enhancer and then to the transcription factor, and it folds it back over here, and then RNA polymerase can bind to the promoter. Okay, and that is actually simplified. It might look something like that in reality, or maybe like this in reality, until you can get those genes transcribed. That's how it works in eukaryotic cells. We don't just have this one control point for a series of enzymes, because we have multiple chromosomes in several different locations, but they might need all the same things in order to turn them on for those particular genes. That's how we can control what enzymes are produced, even though all of them are on several different chromosomes. We then talked about this odd thing about jumping genes, transposons, where they just like self-excite themselves from, just like what happens with the mRNA and the introns and the exons, except this is happening at the DNA level, not at the mRNA level, and the DNA is removing itself and jumping into a different location. We talked about post-transcriptional modifications. The cap, 7-methylguanosine, the tail, 100 to 200 adenines, the poly-A tail. What exons are remaining, how you can modify that. We'll be talking when we discuss the immune system, the millions of pathogens we're protected from just because of variations in 3 genes and how they are expressed. Gives us all of our different antibodies we can have. And here was an example where different exons are left behind. We talked about translational control. Can the mRNA go get out on a ribosome? Is a small interfering RNA inhibiting that in some way? And then last, we talked about post-translational control. Is it folded? Okay, 5 levels of control. Keep them straight in your head. You know, what's occurring at each one. Okay, tell your bio buddy, are you okay on that one? Tell them, yes or no. Do you need to study that a little bit? Okay, now, come back to me again. Then we started talking about the issue that, okay, now we understand all these levels of control, but if your DNA is screwed up, you're going to get a bad protein no matter what, right? Because your DNA is messed up, unless it's a what kind of mutation? Right, and what would be why something could be silent? And that's because for some amino acids, there's multiple ways to get the same amino acid. So let's say you have the wrong codon, but as long as you end up ultimately getting the same amino acid, you're going to get the same protein, right? It's going to fold in the same way. So those are silent mutations. Anytime you're talking about one nucleotide being wrong, that's called a what kind of mutation? Because I can point to that one nucleotide. Does that make sense? That's how I remember it. I point to that nucleotide. And what can end up happening is you could code for the wrong amino acid. You could code for an amino acid or that is hydrophobic when you need one that's hydrophilic, so it messes up the folding pattern. You could code, instead of for amino acid, you could turn it into one of the 3 stop codons. So that's unhelpful. So those are all different ways to cause a problem. A really big way to cause a problem is frame shift. And I gave you an example of codons being 3-letter words. And if you ended up deleting one letter, then all of the codons are off by 1 for the remaining. So every single one of those amino acids, chances are, is going to be wrong. So if you're going to have a frame shift, you'd want it at the very end of your gene, not at the beginning. Because if it's at the beginning, they're all messed up. We talked about that it could be a modification of a protein by various enzymes. And if this one's faulty, you get PKU. But if this one's faulty, you get an albino. But it started with this one. So if we don't have the phenylalanine, we could have both of these issues as well, right? Because it's part of that pathway of modification. we talked about this one, she's XY, but because that Y chromosome is masked in some way, then it's just like she has what? What is it when you're a single X? Turner syndrome. Good. Then we talked about, which we already learned in a previous chapter, we revisited the idea of cancer, and because we know cancer comes as a result of faulty genes, right? Who are the two big genes that we don't want to screw up with that are related to cancer? Proto-oncogenes, which could mutate into, and those are like stepping on the gas. An example of it is gas. Good. What's the second one we don't want to mess up? Tumor suppressors. What is their job? Yeah, cause apoptosis, cause the cell cycle to stop if it's screwed up. As long as you have your ones that can stop, you'll be okay. But if you get an oncogene coding for cell cycle go and you can't stop it, that's when you end up having cancer. And then, what's our example of that? P53. There you go. And then I had some, I don't know if there's a bunch of questions here. You're welcome to try it. You want to, did you get them? Why don't you go in as how many hours of homework you have tonight? How many for me? It's studying, but I didn't assign anything. So technically zero for me. But I'm that's it. 2 and 1 from you. No sleep. All right, here you go. I can go back and do some of the other ones too. I'm going to pause you. Okay, here you go. There's 1. There's 2. Okay, guys, here we go. Okay, guys, real quick, because I have some other ones I want to give you. RNA binds to the promoter. You got that one. Name the part of the operon system where genes are located. The code for the repressor is the regulator. Lactose. Are you okay on those? You know if you missed them and why you missed them? These little questions I think we need to Okay, let's go. Up here, let's do these last 4 together. mRNA getting introns removed. Cut out the introns before it leaves the nucleus. Post-transcriptional. Bar bodies, chromatin structure, right? Bar bodies are an example of what? Thank you. Ability to bind to a ribosome. What are you doing when you're binding to a ribosome? Translation. So that's translational control. 3-dimensional folding. If you're folding a protein, you already have it, so that must have been after translation. Post-translational control, right? Okay, that's it. Make good choices.